Welcome to section 9.4, Translation. In this section, we focus on how the information written into mRNA is converted into a protein. If transcription is copying the recipe from a cookbook, the DNA, then translation is the actual cooking, the step that turns instructions into a working product. By the end of this section, you should understand how ribosomes, transfer RNAs, and the genetic code work together to build proteins. You should understand the three stages of translation: initiation, elongation, and termination, so you can explain what happens at each step. And finally, you should be able to describe the genetic code and how the nucleotide sequence determines the amino acid and protein sequence. Translation is the process of reading the mRNA sequence to assemble a chain of amino acids. It's called a polypeptide. The mRNA you saw produced by transcription is the template. The cell's decoder is the ribosome, and the adapters are transfer RNAs, which we now call tRNAs. And each tRNA has an anticodon that matches an mRNA codon and carries a specific amino acid. Two big ideas to notice here. First, translation happens in the cytoplasm on free ribosomes or ribosomes that are on the rough endoplasmic reticulum. And second, accuracy depends on matching each codon in mRNA with the correct tRNA anticodon. Protein synthesis proceeds in three stages. In initiation, the ribosome assembles at the start codon on the mRNA with a special initiator tRNA carrying the amino acid methionine. In elongation, the ribosome reads one codon at a time, adding the matching amino acid to the growing chain. In termination, a stop codon signals release factors to free the finished polypeptide. These stages ensure proteins are made in the correct order and length. Ribosomes really are protein factories. Each ribosome has two subunits, a large and a small, made of rRNA, or ribosomal RNA, and proteins. The large subunit provides the active site that forms peptide bonds. It's a ribozyme, meaning the ribosomal RNA itself catalyzes the reaction. Inside the ribosome are three lanes called the A, P, and E sites. Transfer RNAs enter at the A site. The growing chain is held at the P site, and empty transfer RNAs leave via the E site. A stands for aminoacyl, P stands for peptidyl, and E stands for exit. This assembly line layout is what makes translation efficient and accurate. Here's the step-by-step guide to how ribosomes work. First, the small ribosomal subunit binds the messenger RNA and finds the start codon, AUG. In eukaryotes, it scans from the 5' cap until it reaches AUG in a good context. In bacteria, a short sequence helps position the ribosome. The initiator transfer RNA carrying methionine pairs with AUG, then the large subunit joins to complete the ribosome. During elongation, a charged transfer RNA, whose anticodon matches the next codon, enters the A site. The ribosome forms a peptide bond that transfers the growing chain to the new amino acid at the A site. The ribosome then shifts one codon. The transfer RNA with the chain moves to the P site. The empty transfer RNA exits via the E site, and the A site opens for the next transfer RNA. This cycle then repeats. Finally, when a stop codon arrives, release factors trigger termination. The chain is released and the ribosome subunits dissociate. Energy from GTP powers several of these steps. The genetic code is the cell's dictionary, mapping codons to amino acids. Codons are three-letter words in mRNA. The key features are that the code is redundant. Most amino acids have multiple codons, but not ambiguous. Each codon specifies only one amino acid. The codon AUG is both methionine, one of the 20 amino acids, as well as the start codon. The codons UAA, UAG, and UGA are stop signals. The reading frame, which is where you start reading triplets, matters. If you shift the reading frame by one base, the entire message changes. That's why initiation is so critical. The genetic code is nearly universal. Bacteria, plants, and humans all read the same codons the same way, with only minor exceptions. This universality is powerful evidence for common ancestry, and it's also practical. It's why bacteria can make human proteins for medicine, like insulin or growth hormone. Another efficiency trick cells use is the polysome. Many ribosomes translating a single mRNA at the same time, allowing rapid, high-volume protein production when needed. Translation converts the information in mRNA into a protein using the genetic code. The ribosome coordinates tRNAs as they match codons and deliver amino acids. Initiation sets the reading frame at the codon AUG. Elongation extends the chain one codon at a time through the APE cycle, and termination releases the completed polypeptide at a stop codon. The universality of the code links all life and enables biotechnology. You should keep an eye out for how antibiotics exploit small differences between bacterial and human ribosomes, another reminder of how central translation is to cell survival.